Hello, we'll start today with an organism that was discovered back in 1890. It's a 100, almost 35 years plus old microorganism. Initially, when it was isolated from grass, it was considered to be a fungus. But over the period of about 130 years, the researcher has now unanimously come to a conclusion that this is not a fungus, it's a, it's a bacteria, and it's a bacteria which is a gram positive. Now, this bacteria, it belongs to the family Actinomycetes, and Actinomycetes are the family of bacteria that form long branching filaments. And these long branching filaments, as we have seen in case of the fungi, they resemble like the hyphae of the fungi. So the long branching filamentous organism resembling with the hyphae of the fungi are the actinomyces. Technically speaking, actinomyces, they are the gram-positive, but some of them, like the Nocardia and the important species of the Nocardia, the Nocardia asteroids, this is weakly acid-fast. So we have two genus that falls into the actinomyces, which have been now classified as bacteria. One is still some of the, you see, some of the mycology, they still consider it as a fungus, but the actinomyces, they have two genus, the Nocardia and the actinomyces. Sorry. No, the actinomyces, they are the gram-positive microorganism, and if you look at the staining over here, but some of the actinomyces, like the members of the genus Nocardia, the Nocardia asteroides, they are weakly acid-fast. Now, if you look into the different species of the Nocardia and the actinomyces and the diseases, habitat, and growth, along with the diagnosis and tabular form, I'm sorry for this. We have the actinomyces Israeli that causes a disease known as actinomycosis. Now, actinomycosis, it is characterized by the abscess formation with draining sinuses duct. And the pus, it contains the, what you call as the, sorry for this. And the pus, it contains the granule, what you call the sulfur granules. So actinomyces caused by actinomyces Israeli, it forms abscess, particularly in the face and the neck, because this microorganism is normally present in the oral cavity and in the throat and the nose. So that's why you see it has the close extremity with the lineage. It forms abscess that really has a sinus tract, and you can see the pus oozing out from this sinus tract, and this pus contains the sulfur granules. The organism is strictly anaerobic. It is gram-positive, and we can see the branching filamentous rod when we culture this microorganism anaerobically. The second important organism belongs to the Nocardia is the Nocardia asteroids that causes nocardiosis. Now, nocardiosis, it forms abscess in the brain and the kidney and the immunocompromised individual. The immunodeficient individuals, it causes brain or kidney abscess, and it can also cause pneumonia. The organism is prevalent in the environment. It's aerobic compared to that of the anaerobic actinomyces Israeli. Again, this organism is gram-positive, but branching filamentous rod, and the culture is often in the aerobic. So Nocardia asteroids is aerobic, actinomyces Israeli is anaerobic. It is commensal in the oral cavity. It is present in the environment. This has sulfur granules. The asteroids, it is present in the immunocompromised. Both causes abscess. Israeli causes abscess in the face area, in the neck area, whereas the Nocardia causes abscess in the brain and in the kidneys. And both are the gram-positive. And if you make a slide from the culture, you can see the branching filamentous rods in anaerobic and aerobic conditions respectively. Now, the actinomyces causes disease, which is known as the actinomycosis. And if you look at the important properties, the organism is an anaerobic microorganism. It is part of the normal flora of the oral cavity, and that's why in most of the cases of actinomycosis, actinomycetes, you will see the lesions in the neck area or onto the face. There is veneer of the tissue occur after local trauma. Whenever there is accidental damage and broken jaws or the dental extraction, being commenced in the oral cavity and in the nose, it has all the possibility to cause an infection in the surrounding area. Filamentous, which is surrounded by area of inflammation. So you see, this is a filament surrounded by the area of inflammation, and this is rich with the sulfur granule. Whenever it will make a sinus over here, you will see the sulfur granules coming out of this. This is a clear-cut, you see the indication of actinomyces infection. Now, clinically, you see the typical lesions, they appears as hard, non-tender swelling, hard, non-tender swelling, particularly in the area of face and area of neck. They develop very slowly, and eventually they drain the pus over here through the sinus tract. So this is how it would look like. You will see this was an abscess, then a sinus formation was there, and this, you can see the sulfur granules over here, along with the blood and sulfur granules. Now the hard yellow granules, what we call that, the sulfur granules, they're composed of a mass of the filaments, and they're formed in the pus. So whenever there is oozing of the pus from the sinus of the lesion of actinomyces, you will have a sulfur granules which are really composed of masses of filaments because of the structure. The filaments, they entangle each other, and they make a mass, and that mass really comes out in the form of sulfur granules. Now, about 50% of the cases, the initial lesions, they are involving the face and the neck, and rest of the 50% cases can proceed to the chest and the abdomen. So lesions always start from the surrounding area, nearby area of the microorganism, the actinomyces israeli. So it causes initial lesions in the lungs and the face and neck, and then move to the other areas like chest and abdomen. Pelvic actinomycosis in women who have really retained an intrauterine device for a longer period of time, they are exposed. This means that the actinomycosis probably also habitat in the vaginal tract of the female as a commencer. And this can occur, and the device is placed for a long period of time to interrupt the pregnancy. They can be affected or infected or with the actinomyces. The most common cause of actinomyces in human is the actinomyces israeli, and there is another subgroup of this microorganism called Arachina, but mostly the Israelis involved. The disease is not communicable, so there is no person-to-person transmission, no aerosol transmission, no environmental transmission. It is just the commensal organism, normally found in the oral cavity and the nose, and that whenever there is an injury, there is trauma, there is broken, there is extraction of the, whenever you manipulate in the oral cavity area by the dentist or through an accident, you can have this infection. Now, the laboratory diagnosis of actinomyces israeli, as we know that they're the branching gram-positive rods, and this can easily be grown onto the laboratory or cultural laboratory media, like the blood agars can be grown also onto the McConkie agar, onto the nutrient agar. This, you will see the branching filaments with the presence of the sulfur granules. The growth from the pus or other tissue specimens under the anaerobic conditions, we take the sample, we plate the sample onto the agar plate, incubate it at normal temperature, and you will have the anaerobic conditions over there to get the growth of this microorganism. The organism can also be identified by the immunofluorescence assay, and there is no serological test available so far for this. So only way left is that we will have to depend on the clinical drainage of the pus from the area, particularly in the face and the neck, and it is having these sulfur granules. And you, if you take this sample, you culture it on the blood agar, you keep it anaerobic conditions, and incubate for about 48 hours time, you will see these specimens having the gram-positive branching rods, and there is no serological test available for this. The treatment is, and the prevention, you see, it requires a prolonged treatment of penicillin G administration, and it must be coupled with the surgical drainage because there is abscess formation. If the abscess is drainage and we don't completely drain the abscess or evacuate this all material from the area, there are possibility of reoccurring of this infection. So what we can do that we can make a incision, can drain all the pus out of this, and we can administer the penicillin G as a prolonged drug factor against this infection. There is no significant resistance has so far been reported to the penicillin G, is quite effective. So you see that even after, because even after 130 years, still the penicillin, discovery of penicillin has also been taken about 60 years back. So still the penicillin is effective. There is no vaccine. There is no prophylactic drug available. We don't need that. But whenever there are incidents of the frank disease appearance, we can count on to the effective penicillin G therapy, and we will have to explore the area to surgically to get rid of the all material enclosed in the lesions. That was all about the actinomyces nizzeli. We have another important organism, what we call as the Nocardia steroides. And it causes disease, what we call as the nocardiosis. Nocardiosis is the disease caused by Nocardia steroids. Important properties compared to that of the actinomyces nizzeli, as we said, that was anaerobic. This organism is aerobic. It is found in the environment. The actinomyces nizzeli was available in the normal form in the oral cavity, and this is particularly found in the thigh. The lung infection in the immunocompromised may disseminate to the other area of the body. It is a gram-positive, thin branching filaments in the tissues, right? Many isolates are weakly acid-fast. Therefore, the normal strength of the acid that we use for the decolorization in the acid-fast stain, that cannot be used in this case. Because if we use the fully concentrated recommended acid for the decolorization of the acid-fast staining, it will completely decolorize this time. And you will find nothing over there. Therefore, the staining process, acid-fast staining process, that is used for the staining of the Nocardia steroid, it has a weaker solution of hydrochloric acid. So it just to decolorize it. So it will retain the dye. The use of the degraded strength acid will decolorize it. So we'll have to reduce the strength of the decolorizing acid compared to that of the standard protocol of the acid-fast staining. Because that's why we see it as a weakly acid-fast. Weakly acid-fast means that it is just required a weak acid to see the fastness of the microorganism against the dye. Clinically, you see there is pneumonia, and long obstructive cavities is a very, very characteristic with this disease. We can see you can see over here the cavity. This is very much. This cavity, whenever you will have a fungal infection like aspergillus, this cavity will be filled with the aspergillus, and it will cause aspergilloma. Whenever you have a tuberculous infection, this cavity will be filled with the mycobacterial tuberculosis. So it causes pneumonia, and lung abscess with cavity is characteristic compared to that of just, you see the infiltration in case of tuberculosis in the lungs. This organism causes cavitation in the lungs. It also causes lung nodules and emphysema later on in the case. So this is how we can tentonograph, what we call the X-ray can give you the clue if there are cavities formation over there, and the organism is an acid-fast microorganism, it's weakly acid-fast organism. There is possibility that it is because of the Nocardia steroids. Now, from lungs, it can spread to the various other organs, and most probably it will go to the brain and will make the brain abscess. Disease occurs most often in the immunocompromised individuals, especially who has the reduced cell-mediated immune response. And we know that in immunocompromised individuals, particularly the HIV/AIDS, they have less than 200 CD4 cell per microliter of the blood. So such a huge reduction in the CD4 cells predisposes the individual to any kind of infection. Even the commensals can become your enemies and can cause a disease. Nocardia brasiliensis, another organism that causes skin infection, and mycetoma. We have another mycetoma that is caused by the fungus organism. Now, the laboratory diagnosis of nocardiasis is it's a gram-positive branching rod. You take the specimen, you culture it, incubate it, and then you see it on the microscope. There you will find the branching rod filaments, and when you stain it with acid fast, with diluted stain acid, it will give the acid fastness. Aerobic growth on bacteriological culture media. Uh, if you look at it, this is how the lesions would look like on the other area of the body. The treatment and prevention, the trimethoprim and sulfamethoxazole, what you call as the Bactrim, the Septron, the other drug of choice, both are still effective. The surgical drainage at some place may be needed, as we have seen in case of actinomyces attest. Rare drug resistance has occurred, and this is the same in case of actinomyces israeli. We still don't have much of the resistance to drug reported against penicillin G. No vaccines, no prophylactic is available. So same line of treatment and preventions, no vaccine available, no prophylactic required, surgeries needed in some of the cases in both areas. The penicillin G is effective in case of actinomyces Israeli, whereas trimethoprim sulfamethoxazole combination is recommended in case of cardiac steroids. I think this is all about the actinomyces. Thank you very much. Hope you enjoyed your time.